Working. Oh, it's working. Now it's working.
I don't know, my laptop, oh yeah, everything's dying. Good, how are you? Yeah. She starts laughing, she's like, that's how it's going to be. Yeah, yeah. Yeah, last spring.
What did they end up doing?
We keep our horses in Tecumseh at a different one.
Oh, okay.
So it was at our family's house, but now it's in Tecumseh.
Oh,
So, yeah.
okay.
They rebuilt the barn, but it's not like an arena or anything. It's just for like their horses, so like the team will always have to be somewhere else.
Okay.
So, yeah, it's sad.
So the place that you're in now, is
I'm not 100% sure. I mean, for this year, I mean, we're there, but I don't know if it's next year or what's going on. 2 hours. I know. I don't know. I don't think anyone knows. I don't think anyone knows yet. So, yeah.
All right. Well, thanks for the update.
Yeah.
Did you have a scrimmage? You have a scrimmage. Oh, I did see a scrimmage day. Oh no, we scrimmaged together with each other.
How's it going?
No.
Well, happy January 30th. I should say happy Friday, January 30th. Yes, Friday is my favorite day of the week, as I have shared before. So I am extra happy today. So how's everyone doing?
Great.
Pretty good, yep. Is Friday your favorite day of the week? Or maybe Saturday, Sunday? I don't know.
Sunday for sure.
Sunday for sure? Okay, good. So, we're doing great in terms of the schedule. We're gonna finish up chapter number 4 today. Then I was able to create a practice exam. So I'll talk a little bit about that and show you where that is on Blackboard. After we finish, like a handful of slides here, and that'll help us to prepare for the exam coming up on Monday. And we'll have more time to talk about that as well. So let's finish up chapter 4 here. So just as a reminder, what's this chapter about? Growth, bacterial growth, very good. And so, we talked about measuring growth in terms of not increase in size, but increases in numbers, right? Yeah. So some of the things that we've been looking at is some of the other some of these requirements for growth, such as what? What do these microbes need for growth? They need
A certain temperature.
They need temperature. They have a certain temperature preferences. What else? Oxygen requirements too can vary. Yeah. Water availability. And then we have fun things like, like carbon sources, energy sources, and things like that. So there's a number of things. And then we looked at, I believe, different ways of growing them using different kinds of media, right? So the last topic for chapter 4 deals with how do we measure microbial growth? So how we measure the number of cells that are present. So this slide informs us that we can do visual counts. You remember looking at those prepared slides of bacteria during our first lab session? They were colored, and then you were able to identify the different shapes. Remember that? Okay. Well, you can imagine you could have counted those bacteria if you wanted to. Yeah, I know they're very small, right? So it's not easy to do, but in concept, yeah, you could actually count each one. So this first graphic here on the left side shows where you can actually use a special slide here. This one's a little thicker than your normal slide, and it's a little bit thicker because it has these depressions or wells built into the slide. So it's in those little depressions there where you can put liquids containing the bacteria, and then you can overlay another glass on top of that has a grid etched into the slide. So what do you think is the reason why there's a grid overlaid on top of the bacteria?
Reference points.
That's right, reference points, so to help you count. So for most people, they're starting at the top left. So you start at that square, you look how many bacteria are present, 0. And then you move to the square to the right. And then so you count the number of bacteria in one row, and then after that you can then move down to the next row below. So the grid pattern is there to help you keep track of your counts. Okay, this special grid is set up so that it covers a certain surface area. So, again, I don't remember exactly what it is, but it'll kind of give you, so that way you can determine like how many bacteria are present in a given area. All right, let's take a look at the next example here. And so in this case, try and imagine that you have like a little laser light source passing through a glass tube, and that glass tube is so narrow, only one bacterium can flow through it at one time. Okay, so that's like super skinny if you think about it. So the bacteria are in a liquid here, and as they're moving down, if they come across the path of that light source, they'll cause the light to scatter, and then you can detect its presence. So every time the light is scattered, you can count. And then based on the numbers, that'll give you an idea how many bacteria are flowing through that liquid in a given volume. All right, so up here at the top of the slide, I indicated that here are some methods where you can count bacteria, but the bacteria that are counted include not only the ones that are alive, but also the ones that are dead. Because when you look at them under the microscope, the live ones look just like the dead ones. They look the same. And likewise, when the living and the dead cells pass through a beam of light, they will both scatter light. So that's the problem with this kind of a method here, is that it doesn't really give you a sense of how many living cells are present, and often that's what we really want to know. All right, so let's move on to the next slide here. So now we are presented with a different method of determining cell counts, and this time we're interested in those cells that are actually alive versus dead. And this method here involves growing bacteria in a liquid medium, often referred to as a broth. So, for example, try and imagine at the top here on the left, you start off with a test tube. It's colored yellow because of the food that's inside of it, and then you can imagine. Let's try to imagine that. Let's say we have, in this example, 50,000 cells per milliliter of liquid here. If you were to take that 1 milliliter with 50,000 bacteria and put it on top of nutrient agar in a petri dish, you would have 50,000 cells multiplying, right? And do you remember what we call those circular masses? Colonies. So you would get 50,000 colonies in one small dish. They would overlap each other, right? And it'd be just this confluent mass. And there would be too many to count. Okay. So in order to count the number of bacteria, what this slide here shows us is that you can make dilutions, serial dilutions. So what this one is showing is that how you can make a 10-fold dilution. So that means you're taking, in this example, 1 milliliter of the liquid containing bacteria, and you dilute it with 9 milliliters of just liquid without the bacteria to give you a total volume of 10. And then you can continue doing those tenfold serial dilutions until you reach a number that you can count in a petri dish. For example, at the very end here, you're trying to imagine that there's 5 cells in that milliliter and will produce 5 different colonies. So because we're only we're only interested in live bacteria, the live bacteria will multiply and form colonies. The dead bacteria will not. And so by counting the number of colonies, they'll give you a good estimate of how many live bacteria are present. And then because you know how many tenfold dilutions you made, you can then calculate the original concentration of that liquid based on the number of colonies observed in that final dilution. Okay, any questions about this one here? Okay, looks good. All right, so again, you can grow bacteria on solid media, like inside of a petri dish. You can also grow bacteria in a liquid state, in a broth, for example. Like in this test tube here, let's say we have bacteria inside of in 1 milliliter of liquid in a test tube. And then you can transfer that liquid into the petri dish in one of 2 ways. So on the top, we have what they call a spread plate method. So you basically just put the liquid on top of solidified agar medium, and then you can take this glass rod and spread that liquid out over the surface, and then the living cells will multiply and produce colonies, which is what you see here on the right. Okay, so that's like the the most common method. But another way you can do this is to actually take liquid containing the bacterium from your test tube on the left and actually add it to melted agar containing your food items in there, and then pour it into an empty petri dish. And then when that agar solidifies, you'll have bacteria not only at the surface, but also within the agar itself. We're actually going to do this. We're actually going to do this method here at the very end of the semester, when we're working with viruses, because viruses can't replicate on their own. They actually need a host cell. So we'll actually get some viruses that can infect bacteria, and we'll actually combine them together like what you see here, using that pour plate method. So just kind of put that one in your memory banks for until the end of the semester, because we'll pull this one back out. Okay, here's a fun term there that I don't know why we have so many terms for the same thing, but so we understand that a colony is a mass of bacteria that you see growing in a petri dish, and they originated from a single cell, and that cell or that colony kind of gives us an idea of how many of those original cells are present. And so someone came up with the idea of calling that original cell a colony-forming unit. So this is like the original cell that was present in this liquid here that gave rise to a colony, a colony-forming unit. All right, any questions about this slide here? Okay, let's keep going. All right, so remember that we're learning about different ways of measuring or counting bacteria. So another method involves using a filter. So what we're looking at here is a membrane here that has very small pore sizes. Does anyone remember what are the average sizes of a bacterium?
5 micrometers.
Yeah, about 5 or in that 2 to 5 micron or micrometer range. That's right. So just in general, that 5 refers to this length here. Okay, this is about 1 from left to right here. So this is 1. This is roughly 5. So knowing that. Those are micrometers or microns. How big do you think this little pore here is? What do you think? This little hole in the membrane. If this is 1, across from left to right on that bacterium, how big is that pore? It must be less than 1, right? So what do you think?
Quarter or half.
Yeah, about 1/4 to half, exactly right. Exactly right. So can you imagine trying to make those tiny little holes? But I think you can kind of see these holes allow liquid to pour, filter through, flow through, but they're small enough to retain microorganisms. And then the idea is that you can take those filters, put them on top of a petri dish containing a nutrient agar, and then count how many colonies form. Now this example here shows how you can take a source that contains, like water. It could be drinking water. It could be untreated water from a reservoir or a stream or a pond or something like that, and you can test how many bacteria are present. For example, you probably have heard examples of where swimming beaches have been closed. How come? Well, maybe E. coli, there's E. coli outbreaks. Like E. coli is what you find in human feces and waste, sewage, overflows and stuff like that. So if you find E. coli, that's an indicator of that kind of stuff. So like how do they know? Well, they can actually take water from that beach, filter it, start counting them, and then see if the numbers are above, like normal levels or not. So. But wait, you can also use this filtration method not only with water, but with what? You can not only remove bacteria or collect bacteria from using filters from water, but also what else? Gas, that's right, air, right? And remember we had that one laminar flow hood in the laboratory that we're using when we're taking out our petri dishes out of the bag? That's got a HEPA filter. So the HEPA filter is set up like this one here. And I don't know if this room has any filters. It may or may not have some filters there. Hopefully, hopefully it's trapping or collecting some of those microbes so that we don't have to constantly inhale them. So that's another So don't forget about the air filters too. Your textbook mentions this most probable number method. So this is just like, this involves serial dilutions like we've seen before. And there's actually some, like instead of making all these tenfold dilutions all the time, I mean that's basically what we're looking at here, tenfold dilutions. But then the idea is that someone actually worked up a table to tell you that, if you find a certain number of bacteria present in a particular dilution, they'll tell you using probability how many bacteria were present in that original solution. So this is just like what we've seen before in concept. And then I think we're just looking at a couple more slides here. You can also measure the number of cells based on mass. So that's their weight. So people typically don't do that with bacteria because bacteria are just so small. But try to imagine if you're growing fungi. They're much bigger. You can then grow them, harvest the fungi because they're so big you can actually see them, and then dry it and then weigh it. And then the more cells that are present, the more it'll weigh. And then the other way here is using turbidity. So remember in that cell counter that used the laser beam that if cells are present, they'll refract light. So that's the concept here, that you can actually use a special instrument called a spectrophotometer to measure both transmission and absorbance of light. And we'll see the example here in the next slide. Okay, let's focus our attention on these two test tubes or glass tubes here in the center here. So one of them has bacteria and one of them doesn't. So which one do you think has the bacteria? The one on the left, that's correct. How do you know that? Because it's all cloudy, right? Compared to the one on the right, that is clear. Clear doesn't mean transparent, right? Because this one, everything's got a tint of yellow because of the food that's present in the tubes. Clear means that light can pass through, so you can actually see the other side if you look through this tube here. But then if you look here on the left, because of all the bacteria that's present, that prevents light from passing through. And then the idea is that we can quantify that using a spectrophotometer. So I think we have one of these in the lab here. So basically, have you used one of these in chemistry class, a spectrophotometer? Yes? Okay. There you go. So the tube actually goes over here. This is our tube holder here, and then you can set the absorbance, the wavelengths that you want to look at. And in this little graphic here on the upper right, if no bacteria are present, light from the light source will pass through, and so you'll have high transmittance, low absorbance, and then you can measure that numerically. But here on the bottom right, if you have a lot of bacteria present, the bacteria will absorb the light, but then you'll get lower transmittance. And you can record the values either in absorbance or transmittance here. Okay, so that'll kind of give you like an indirect way of determining how many bacteria are present. And you can actually, build these standard curves where you can actually quantify the amount, correlate the amount of absorbance with a certain number of bacteria, a certain concentration of bacteria that are present. All right, any questions about this one here? Good. Okay, I think this is our last slide here in the set. We already talked about how you can measure the weight, so that's like biomass. But let's focus our attention on the last part here, and that is you can look at the products made by the bacteria and use the products, the amount of products is like a proxy for the amount of cells that are present. So we have a couple of examples here. So we're actually doing this one as well. So in about 4 weeks, we're going to take your bacteria and we're going to test it for glucose and lactose fermentation. So in other words, can they use glucose and lactose, which are sugars, can they use that as a source of food and ferment it? And the way that this one is set up is that we'll have these like test tubes. So try to imagine that this drawing here is a test tube, and it's going to be filled with liquid nutrient broth. And then inside of this thing, we have some pH indicators. So what does that mean? What does a pH indicate? What is a pH indicator?
Changes color when the pH changes.
Exactly, that's right. All right, so we're gonna have bacteria in here, but then we're gonna add a tiny tube called a Durham tube. And on top of that, it's gonna be inverted, it's gonna be upside down. So we're gonna have inside of this tube, we're gonna have a smaller tube. That's our Durham tube. Okay, so we're going to have bacteria. We're going to put your bacteria in here. We're going to put, glucose or lactose. That's sugar. And then we're going to see if it can ferment it. So, can it make acid? Can it convert this into an acid? And how do we know? Well, then we add the pH indicator in here. So if we get the acid, we're going to see a color change. Okay. So the more color change we have, and the more acidic it is, that means there's more bacteria present. So that's how we're using cell product as an indicator of how many bacteria are present. And then we have CO2 production. So let's say our bacteria make gas. What will happen is that some of the gas is going to get trapped inside of our tube, and so it'll look like a little gas pocket inside of this liquid. And that'll give us an idea of how many bacteria are present as well, based on how much gas is in that tube. All right, you guys are learning a lot. Again, when I was your age, I didn't know anything. All right, any questions about this stuff? All right, we have some time here. Let's take a look. Let me show you the Blackboard site. Here's the Blackboard site. Okay, so this is our Blackboard site. Okay, I don't know if I can make that bigger or not. Okay, there we go. Okay, so under the course information, there's our practice exam down here. So I don't know if you have your laptop with you, but you're invited to kind of look at it along with me at the same time, so we can kind of look at this one together. Okay, I think I had to download this one. I'll open it up and let's take a look at how this one is formatted here. All right, 100 points. So you're gonna get a paper test on Monday that looks, formatted the same as this practice test is here. So what kind of questions are we looking at, or what types of questions are we looking at here? Fill in the blank. Okay, let's take a look at this first one. Using a blank. Then Leeuwenhoek and Hooke, Robert Hooke, were the first persons to observe microorganisms. What do you think? Microscope. Okay, good. It's like read my mind. Good job. All right, so you're gonna see some of these, fill in the blanks. We also got questions like, like short answer kind of stuff. Matching, so that's what we're looking at here, like matching terms. More fill in, short answer. Fill in the table here. Circle and underline. Okay, we got a little. We got some numerical answers too. So, like, can you convert, like different units? So, like this symbol here. What is this symbol here? What is this one here? That's our micrometer. That's right. Anyone remember how many micrometers are in a meter? It's 1 million. 1 million. So 1 micrometer is 1 millionth of a meter. Okay. So don't worry, you have a weekend to study. Okay, drawing. Can you draw shapes and arrangements? Okay. More matching. Oh, labeling. We like the microscope. We need to be able to identify the different parts of the microscope. Yeah. Okay, I also think, okay, how about true false? You like that? All right. All right, and so those are the kind of short questions that we're looking at here. And I'll just briefly describe my philosophy of teaching here. I don't know, like in driver's ed, when you learn how to drive, did you take a class? Did you go through driver's ed class? Okay, I did too. And in my driver's ed class, we had some reading materials, you can take home with you, learn how to drive. I had an instructor kind of giving, short lectures. Watched a bunch of crash videos. Those are fun. But then after that, what did you guys do? Drive, that's right. You got to get in the car and practice. Practice your learning. And that's the concept here that we're working on. You know, you're presented with lectures, you're presented with your textbook so you can read the information. You got the PowerPoint slides, you got your notes. You know, we got the recordings in case you want to go back and listen. Okay? But then you need to practice your learning. So that's the idea behind the practice test here. Kind of gives you an idea of what you're going to be tested on and what you're going to be expected to know. So in other words, you know, like when you in driver's ed, did you have to practice parallel parking? Because that was on the test. At least it was on my test. So before you get tested, you practice. Practice parallel parking, and then you get tested on parallel parking. So what you're looking at here, this is the practice before you get tested. So you're going to see very similar questions on Monday, because you get to practice before you get tested. Now, similar kind of reasoning here. Does it make sense to practice parallel parking and then you get tested on backing up a trailer? No, that makes no sense. And as a former dean, I used to get so many complaints from students who would complain about their professors. You know, they get exam questions that have nothing to do with what they practiced. You know, there was that mismatch here. But so I'm very aware that you know how learning and assessment should work here. So take the practice exam seriously. You know, learn from it, and use that also as your means for studying to prepare for Monday. All right. I actually aced my my driver's ed exam. Anyone else do that? Good job. All right. Any questions about the practice exam on Blackboard or what's going to happen Monday? Yeah.
Is there any sort of key for the exam, or is that on us?
Yeah, I think you'll be able to figure that out. You know, we did the first one together. I mean, okay. True or false? The aseptic technique is used to prevent contamination.
True.
That's true. Yeah, like we did the hand washing, right? We had the gas burners going. We're using the disinfectants, all to prevent microbes from entering into our clean petri dishes. Yeah. So, yeah, I mean, you should be able to figure these things out, right? But if not, you always have the internet. Okay. Other questions? Okay, don't forget, your chapter notes are also due on Monday. So I think most of you are, you're on pace, you're on track, you're turning those things in as you go. So I just want to remind you about that. Monday's the deadline for that as well. So. Okay, so bring your lucky pencil with you on Monday, and I will see you then.
So that was just a big 2 by the edge. I think that for you. I was like.